Next, I would like to welcome Dr. Andrew McKenzie, the Director of Personalized Medicine at Sarah Cannon Research Institute. Dr. McKenzie earned a PhD in pharmacology from the University of Vermont, followed by postdoc work at both the University of Vermont and Vanderbilt University. Dr. McKenzie and his team are the go-to experts on understanding molecular profiling testing, understanding results, and helping match patients to the best trials for them across the Sarah Cannon Research Institute network. We're thrilled to have Dr. McKenzie join us to discuss personalizing melanoma treatment for patients with stage 3 and 4 disease. Please welcome Dr. McKenzie.
Thank you so much, Dr. McCain, and thanks to everybody here for joining. I'm delighted to be here and excited to talk to you today about personalizing melanoma treatment for stage 3 and 4 patients. I'm going to start some slides here in a second, but to reiterate on what Dr. McCain said, my team here at Sarah Cannon is really involved in understanding what we call the basic biology of tumor cells. So all of the excellent analogies that Dr. Falchuk just shared, it's our job to understand the nitty-gritty science behind that and to try to link the complex genomics and genetics that are happening inside of a tumor cell to what is an ever-growing list of drugs that can be given to cancer patients. And we call that matching of what's going on inside of a patient's tumor cell itself to what drugs we have. We call that matching personalized medicine. And this time, 10 years ago, as Dr. Falchuk mentioned, this really wasn't possible for a lot of cancer patients. But now with the onset of new technologies and the great work that folks like Dr. Falchuk, McCain, and others are doing, we have incredible access to cutting-edge technology for more and more patients, and then access to these wonderful novel therapies so that we can truly personalize cancer care. Again, I'm delighted to be here. Thank you so much for having me. And I'm going to share a few slides. And I don't think I can live up to Dr. Falchuk's analogies or his voices, but I'm going to try my best. It's going to be a tough act to follow, Dr. Falchuk. Hopefully you can see my slides here. If you can't, somebody yell at me. We'll get started. So for today's talk, we're going to quickly go through what we consider to be the foundation of cancer itself, the fact that it's a disease of the genome and what that means. And to do that, we're going to get a little bit nitty-gritty in how cells work and what goes wrong when they don't work correctly. And that's going to lead us into talking about all of the different targets or biomarkers that are present in melanomas. Dr. Falchuk's example of BRAF is one of many different targets that we now have that we can target in melanoma. And then lastly, and I think most importantly, how we test for these biomarkers. What the difference is between things like germline genetics versus somatic genomics, how to get results from blood-based testing versus tissue-based testing, and what all of that means for the cancer patient. Hopefully by the end of this, we'll learn together that though this is a pretty complex process, that all of this is available today for cancer patients receiving their care. So we'll start with cancer as a disease of the genome. And what I mean by that is, at its most fundamental level, you can trace cancer back to something that's gone wrong inside of a cell. So if we're going to take a journey back, and hopefully that'll make us it easier to go forward here. But if everyone will remember from high school biology class that organisms, we humans, are made up of organs and organ systems, and those are made up of tissues, which are made up of cells, and inside of those cells, you've got these subcellular machines that we call DNA and protein and RNA and others. And these are the things that are responsible for making cells do what they do. And so we as cancer biologists and scientists are very interested in how things work at this molecular level and cellular level, because everything else is based upon what's happening at these molecular levels. So bear with me as we go into what these tissues, cells, and subcellular machines like DNA and protein, what they do and what they look like. So this is my analogy for the day, and it's, I think we scientists like to try to contextualize things to make sense of them. And so the best way I know how to make sense of what a cell is it's like they're these little bitty molecularly sized factories. And this helps me try to understand the different pieces that make up a cell. So imagine the cell being the factory. Inside of the factory, you've got these things called proteins. Those are the machines, they're these things actually doing all the work inside of the cells. And then you've got the DNA that everyone's probably heard of, and we consider that to be the blueprint. You've probably heard that term before, that the DNA is the blueprint. Well, this is what it means. If you're building a factory or have one, the blueprint tells you where to put all the machines and how they're supposed to work. The proteins are the actual machines, and you put it all together and you've got yourself a factory. So what happens at the factory and what machines are present? This is really where we get into what targets are available. Dr. Falchuk shared that we have these wonderful inhibitors of things for BRAF and others, and I'm going to attempt to show you how those work and what goes wrong when we have these mutations that pop up. So protein machines that exist in the cell, we've got several different types. I'm going to focus on three main ones just to orient everybody. We've got these ones that Dr. Falchuk referred to that sort of stick out through the cell membrane and out into what we call the extracellular space. And what they do is interpret everything that's going on around them. There's little antennas that stick out. And these are primarily responsible for integrating what we call growth signals. So they sense that it's time to grow, and then they capture that signal and then propagate it down inside, and then the cells start to grow. This happens as a normal part of development. All of our cells are tuned to grow and then stop growing when they don't need to. And these, what we call cell surface receptors, are responsible for primarily getting that cell and translating it into these intracellular signals. Things in melanoma that you all might be familiar with that fit this bill are things like the KIT protein. And so it sticks its hand out, binds to growth factors that are present, propagates those signals downstream, and tells the cells to grow. In normal cells, this is in balance with growth suppressors so that we get just the right amount of growth. Next, on the next layer in, so you've got these receptors on the outside that bind to and grab these signals, and then the next layer down is what we call the signal transduction pathway. It's a fancy way of saying, how do we take that and, like the quarterback, pass it to the next person that's supposed to do something? And this is where you see things that are really common in melanomas like BRAF, KRAS, NRAS, MEK, and others. So these sit just inside the cell surface and are responsible for taking that growth signal from the receptor and propagating it down into the cell. And so all of these pictures I'm showing on the right are actual pictures of what these proteins look like. So we've been able to isolate them and take very high resolution pictures of them, and this is actually what they look like. It's pretty amazing. And here you can see things like KRAS and BRAF and MEK and others. And this is how we're able to design drugs that target these, because we know exactly what they look like, and we can build drugs that look like binding pockets inside of these. And then the next layer inside, as you're getting deeper and deeper, are these proteins in the nucleus of the cell. And these help to bind DNA and control synthesis of other proteins. One very common one is the estrogen receptor, and these are responsible, again, for taking those growth signals. They get propagated down through these cell propagation or signal propagation patterns, and then into the nucleus through these proteins to actually do the communication in the nucleus, to grow more and grow more. And again, I'll stop with, or finish here with what we call the central dogma of molecular biology. I know I'm getting into the weeds here, but this is really critical to understand how these mutations can result in something that's actually targetable on a clinical trial or with a drug. So the central dogma says, again, the blueprint, the DNA makes everything. The DNA makes RNA, makes protein. And if something goes awry in the DNA, then that is made manifest inside of the protein. So really, when we're talking about these genomic sequencing and how to find these mutations, we're really talking about surveying the DNA to see if anything's gone wrong there, because we know that if something has gone wrong in the DNA, it's going to make itself manifest in the protein. And I've got a few examples of what that looks like. So under normal circumstances, everything is fine. The DNA looks normal. You make a normal protein. We call that wild type. It's what's found in nature. That's normal, no mutations present. In a lot of cancers, we can get these mutations that arise, and melanoma is no exception, that cause a specific defect in the DNA that's made manifest in the protein. One example here that I have on the screen, BRAF V600E. So at a very specific place on the BRAF protein, part of that machine was made incorrectly. You can think of it as in the factory, there isn't a bolt where it's supposed to be, and so now the machine is going haywire. Similarly, you can have situations where the entire protein isn't made. This is where you make a certain amount of it and then it stops. And again, if you've got a machine where you've only got half of the machine made, it's not going to be doing what it's supposed to be doing, just another type of mutation. And then lastly, we get these really weird things called fusions, where two different proteins get fused together and create one unwieldy protein that's made of two different proteins. And these are called fusions. And what happens at the genomic or genetic level is you've got two different genes that get fused together. And what that does is make and manifest a fusion protein. All of this is important because each of these types of mutations can be targeted with specific therapies. So as I mentioned, everything is in balance in a normal cell where you have that growth factor receptor that says, hey, it's time to grow. And then there are growth suppressors, or what we call tumor suppressors, that say, all right, it's time to stop growing, and it keeps everything in balance. What happens in a cancer cell is that one of those things gets out of balance. You've either got way too much growth signaling going on, or not enough growth or tumor suppression going on. And that's what caused this sort of uncontrolled cancer growth. And the reason that you have that imbalance is typically because of a mutation that occurs in one of those two places, a mutation that causes the machine here to go into overdrive, or a mutation in the machine where you can't pull the brakes. You can't stop it from creating more and more product. So that's, in a nutshell, what goes wrong with cancer cells. And we use that knowledge to go and find different mutations that might be sensitive to inhibition or sensitive to drugs in the clinic. There's a lot of discovery, as Dr. Falchuk mentioned, that goes on in the laboratory to try to find these mutations in cancer that cause this uncontrolled growth. And when we do find those, we then take that into the development pipeline and say, how can we create a drug that inhibits that mutation, inhibits those proteins from causing this overactive growth? So we're going to switch gears a little bit to start talking specifically about those targets. What biomarkers have we identified in melanoma beyond BRAF that we heard so wonderfully about earlier? And I'll be really upfront. This slide's very busy, but I think that's an encouraging thing because that means that we've got lots of targets that we could talk about. Hopefully the pictures on the right remind you of slides you just saw. Because we do have targets at sort of each level here, sort of those cell surface receptors that are finding growth signals that turn them on. And then also this sort of middle level signal transduction group of proteins all the way down into the nucleus. All of those exist inside of melanomas. And so, for example, we've got things like KIT. I showed you a picture of KIT earlier. There are specific inhibitors like imatinib and others that have been developed that specifically target mutated KIT. EGFR is another one. We've got things, of course, like the BRAF and MEK story. Interestingly, we have things like NRAS where we don't quite yet have a grasp on how to target NRAS, but we do have clinical trials that are looking for folks who have NRAS mutated cancers, trying to see if this next generation of drugs can work in this patient population. Things like PIK3CA, ALK, CDK4 and 6, and others. These are all targets specifically in melanomas that can be looked for to see if there are mutations in these genes. Additionally, as Dr. Falchuk mentioned, we've got a whole host of immunotherapies that have come into the clinic. And these immunotherapies also look for the presence of specific biomarkers or proteins. And it's knowing that the presence of these biomarkers are there that we can target specific types of immunotherapies for some patients and not for others. Again, it's the whole concept of personalizing treatment based on a patient's genomic profile and their immunotherapy markers. So these letters and numbers here, CTLA4, PD-1, PD-L1, IDO, and so forth, are all markers that exist either on the tumor cells or on these T cells that Dr. Falchuk talked about that give us a sense of whether or not a patient might respond to a particular immunotherapy. And again, I know this slide is busy, but that's a good thing because it means that there's so many different areas of investigation that you can see here for different classes of immunotherapies that are under investigation, including viruses, oncolytic viruses, specific antibodies that we can put inside of a cancer tumor environment, cell therapies, and others. I think this is just such an encouraging and exciting landscape to be studying these days. So then the natural question is, okay, Andy, you've taught me or shown me all of these different ways that something can go wrong in a cell, and you've shown me how that's bad, and all these different targets that we know exist. But how do we test for those? How do I know if I have one of those? What actually goes on behind the scenes so that I can know if one of these is present in a tumor that I might have? So the first thing, I'm going to flip through here to get everything on the slide. The first thing to talk about is the difference between germline mutations, what we call germline or inherited mutations, and somatic mutations or acquired mutations. So you all might be familiar with things like the BRCA gene in breast cancers and ovarian cancers. These are inherited mutations that are passed down through germ cells and are present in every cell in your body. These are really important for the diseases that I talked about, like breast cancer and ovarian cancer and others. But most cancers that we deal with, and particularly the markers that we're concentrated on in melanoma, are ones that are acquired somehow, whether it be through UV exposure or from, you all probably know, the smoking story in lung cancer and these carcinogens that propagate these mutations and that cause overactivity of cells and cancer. So in the world of melanoma, we focus mainly, but not exclusively, but mainly on the somatic side, this acquired side. Not as much of the melanomas that we see, not as many of them arise from these inherited or germline mutations. So in somatic testing, which I'll focus on here on the left, you get information about the tumor mutation pattern a couple of different ways. The first is through a biopsy, by taking a literal slice of the tissue and taking that and then doing the lab work. The other one that has come into a lot more prominence recently is this cell-free DNA, or a blood-based assay. And you can see in the middle of the screen here, this one syringe that is inside of the bloodstream taking very small portions of DNA that we call cell-free or circulating tumor DNA. And we are able to sequence that DNA and find mutations. And this is DNA that's shed specifically from tumor cells that's in the bloodstream. And that's different than the germline testing where you're actually taking the DNA from the white blood cells that are in the blood or from a cheek swab. This is actually taking DNA that's been shed from a tumor. And so these two different ways, either from a solid tissue biopsy or a cell-free DNA sample, can give you insight into the mutations that might be present in a tumor. So what happens then? After you take the biopsy, this gets sent to a laboratory who actually does the sequencing. And what that means is these are highly trained professionals who are scientists that run your tumor's DNA through a sequencer, and they're able to sequence out all of that blueprint, the DNA, and then compare that to a normal or non-mutated DNA and see if there are any differences, and then capture those differences, put them in a report, and send them back to people like Dr. Spalchuk and McCain and others. And then that's where they do the great work of matching those mutations to specific therapies. And I know that I'm kind of blowing through these last few slides here, and I'm coming up on my time, but this is really it in a nutshell here. You see mutations in things like BRAF, NRAS, KIT, VDR, and others at fairly high frequencies in melanomas. And this is all the more reason to get these tumors tested to see if those mutations are present. This slide here, I think, does a good job on the right here of saying how cancer therapy is evolving. It used to be in traditional chemotherapy that if a person had melanoma, they would get a specific cancer treatment. As Dr. Spalchuk mentioned, now there are different types of melanoma that are molecularly defined, BRAF mutant melanoma versus NRAS mutant versus so on and so forth. And depending on what mutations are present, that means that different medications are more efficacious and better in those. And I'll say this until I have no breath left. You don't know if a molecular alteration exists unless you test for it. And you test for it through the mechanisms that we talked about on the last slide. And so I'll encourage folks in my last couple minutes here to speak to your doctor about this. Use the terms next generation sequencing. Have you sequenced my tumor? And know that a lot of these targets that we talked about, and I'll flip back 2 slides here so you can see, a lot of these targets are only available to be tested through mechanisms like next generation sequencing. And a lot of these targets are what's being used to stratify patients to get them on clinical trials that Dr. Spalchuk talked about. So I'll encourage everyone, talk to your doctor about that. If you haven't had an NGS test, talk to them about that. And I highly encourage that, and we'll stop there. I want to thank again you all for your attention. I know I got into the weeds a bit. I appreciate you coming along for the ride, and I'm grateful to have been a part of this today. Thank you.
Great. Thank you, Dr. McKenzie. That was fantastic. I think a lot of our patients that are listening in are probably taking some notes, some questions back to their doctors about, have I had this testing done? If so, what does that look like? So I think in my experience, our melanoma patients are very well informed. So hopefully they enjoyed your presentation as much as we all did.